Hi everyone, today we're looking at trinucleotide repeat expansion. Occasionally, a sequence of three bases, that means codons, that is repeated in tandem will become amplified in number so that too many copies of the triplet occur. If this happens within the coding region of a gene, the protein will contain many extra copies of one amino acid. What are they trying to say? So basically, normally, uh, what do you call this? The mRNA, it is, wait, let me change the pen. Yes, so the mRNA, it is structured some way like this. It has a 5 cap, then it has a 5 UTR, which is untranslated region, then it has a coding region, then it has a 3 untranslated region, 3 prime untranslated region, then it has a poly A tail. And this is the 3 prime end and this is the 5 prime end if you talk about it. So normally, if they're saying that codon are basically made up of three nucleotide bases, right? So normally they will be repeating, which is fine. But if they start repeating numerous number of times, especially in the coding region, so then this means that there will be a lot of certain proteins that will be produced, and this can be very problematic because it can result in various diseases, including Huntington disease. Then there's myotonic dystrophy. There's Fragile X syndrome. But if so, Huntington disease, this takes place when there's additional number of those proteins. But if there is decreased number of those amino acids, then there'll be myotonic dystrophy and Fragile X syndrome. I'll explain this to you line by line. So let's look at this. Trinucleotide is basically what? It's a three base. So trinucleotide is that three bases, the three nucleotide bases, right? So these are three nucleotide bases. This is one codon, and this is the other codon. This is the third codon. So what do you mean by three bases? So one base, two base, three base. So that's one, like that's one entire package of trinucleotides. And again, there's a trinucleotide. There's another trinucleotide. So trinucleotide repeat expansion. That means that this will keep on repeating. Trinucleotide repeat expansion. A short three base DNA sequence is repeated too many times. So this is the definition that should be on your fingertips, that a short three base DNA sequence is repeated too many times. So short three base DNA sequence. Then moving forward, normally genes have a small stable number of these repeats, which is fine if there are a few number of repeats. In disease, the repeats increase abnormally, but in disease situations, the repeats will be increased abnormally. What happens when repeats expand? It depends on where the expansion occurs. So on the location is the problematic part. If it's in the coding region, then it's going to be problematic. But if it's some UTR untranslated region, so it can be bearable, it can still be manageable. Expansion in the coding region results in Huntington disease. So normally there are 10 to 30 CAG repeats. So this is a trinucleotide. So normally 10 to 30 repeats of it are fine. But in cases of diseased condition, there'll be more than 36 CAG repeats. And CAG codes for glutamine. So this means there'll be extra glutamine residues. So this is what they're saying. Result would be that protein has too many glutamine residues. Protein becomes abnormally long because there's so many unwanted repeats. It misfolds and aggregates. So there'll be misfolding and aggregate because the chain of the amino acid will be abnormally long. So it will start misfolding and aggregating. So aggregates will form and it can cause neuronal death. This explains the neurodegenerative diseases. So that's the reason. So this explains that this is a neurodegenerative disease. The reason being that there were excessive glutamine, a lot of amino acids. This results in abnormally long amino acid chain. When obviously there's a long chain, there are chances of it misfolding and aggregates forming. When aggregates form and they accumulate in brain, they'll be result in neuronal death, and that's the reason this is neurodegenerative disease. And here also you can see Huntington disease. So this is the 5 cap and then 5 prime cap, and then you have the poly A tail. And this is the 5 prime UTR, this is the coding region, this is the 3 prime UTR. So this is the mRNA structure, right? Now, tandem repeats of CAG triplets coding for glutamine. So glutamine amino acid is coded by CAG, so this is the trinucleotide. So there will be a lot of repeats of it, at least more than or equal to 36. And mRNA That mRNA, it will be translated, obviously, and there'll be a protein forming. That protein will be Huntington protein. Huntington protein is a protein which is an abnormal protein which has extremely increased numbers of glutamine amino acid residues. So at least 36 or more than 36. So this is an abnormally long amino acid chain, and obviously there will be misfolds and aggregates will form. It will result in neuronal cell death, and ultimately it's a neurodegenerative disease. Aggregated proteins in Huntington disease, a polyglutamine disease. Um, Expansion in untranslated regions. If there's expansion in untranslated regions, here the protein sequence may be normal, but gene expression is affected. So even though the protein sequence might be normal, the gene expression will be affected. For example, Fragile X syndrome, where there is CGG repeat in 5' UTR. Myotonic dystrophy type 1, where CTG repeat in 3' UTR. So trinucleotide repeat, but in this case it's 5' UTR in the Fragile X syndrome, and in myotonic dystrophy type 1 there is 3' UTR. So initially I mentioned that there are decreased amino acids. I was wrong. Instead, they're telling here that the repeats are in different locations. So depending or based on the location where the repeats are happening, diseases differ. Result would be abnormal mRNA handling, gene silencing, or faulty regulation, disease without abnormal protein structure. Why do repeats expand? Okay, so this is another diagram which is other triplet expansion diseases. Fragile X syndrome in the 5' UTR region, there are repeats of CGG more than or equal to 200. And in myotonic dystrophy, in the 3' UTR, there would be repeats 100 to 1000, right? So yeah, if you want, you can further read this. Here they're saying that a Fragile X mental retardation protein, it is 5 to 44, and in myotonic dystrophy protein kinase, it is 5 to 34. Okay, moving forward. Why do repeats expand? During DNA replication, repeats cause DNA polymerase slippage. Extra repeats are added. What does this mean? This means whenever the DNA is replicating and then only RNA will be formed, right? So during the DNA replication, that's when the problem occurs. There is DNA polymerase slippage and extra repeats are added. So there are normal repeats, but then because of those repeats, the DNA polymerase will slip and there will be abnormal amounts of repeats added. This is unstable across generations. What does this mean? This is a very important line. But this is unstable across generations. This means that it will result in anticipation. Anticipation means that the repeat number increases in successive generations. Very important. For example, the grandparents, if the disease is there, so it won't be a very severe form, right? But later on, it's going to develop into the extreme case of that disease, right? This phenomena is called anticipation. This is seen especially in Huntington disease, Fragile X syndrome, myotonic dystrophy, these are the three examples we've discussed. Anticipation means a genetic disease appears earlier and becomes more severe in each successive generation. This is the chart that you can see. Grandparent had 40 CAG repeats, but in parent, the 50 CAG repeats. So with the successive generation, the repeats increased. In the child, the CAG repeats were 30. So severity of the disease would obviously be more in the child who has extreme number of repeats. So this progressive worsening is anticipation, and anticipation is seen in Huntington disease, myotonic dystrophy, Fragile X syndrome, spinocerebellar ataxias. And many genes contain trinucleotide repeats. Expansion can disrupt protein structure, transcription, mRNA processes. So these are the diseases. So yes, that's about it.